So we're going to be going over the second half of day 5/day 6, the beginning of life. And last time we ended off with the teleological argument. Now this is the argument that basically is the design argument. So looking at things and seeing that they have evidence of design, which implies that there is a designer. Now, we don't actually have to see the creation process or the creator itself to know that something has been designed. God has shown man the truth, such as Romans 1:20, for the invisible things of him from the creation of the world are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse. So we can see everything in life, both biological and non-biological, has very fine-tuned design components. And that is a theistic explanation for what we see. Yet, here in the last 200 years, there is an anti-design, anti-theistic explanation. And that is that adaptation happens by random mutation and natural selection. And that is basically whichever adaptations allow the organism to grow, metabolize, and reproduce the most efficient, survive, and outcompete the competitors. As Peter Godfrey Smith says, the apparent design of organisms and the relations of adaptiveness between organisms and their environment are the big question. The amazing facts in biology. Explaining these phenomena is the core intellectual mission of evolutionary theory. Natural selection is the key to solving these problems. Selection is the big answer. Yet, relying on natural selection as the new designer, even though it cannot make any true choices, is the denying of the idea of a designer at all and denying the idea of God. So to go through some of these, we're going to start with the cellular structure. Now this is a eukaryotic cell. There is also a prokaryotic cell that is a bit less compartmentalized, meaning that there's not these little rooms inside the cell, but it is by no means not complex. We're just going to go over this because these are the kind of cells that we have within our own bodies. And we're going to start off with DNA. Now DNA is made up of 4 different base pairs. Just remember A, T, and CG. And this holds a ton of information, kind of like Morse code on a string. You can see an example of this in the bottom right corner. Many of you right now may be actually saying, help, with all this information. Now the key thing is you don't have to remember all this. This is basically a semester, if not 2 semesters, in a college level biology class. And we're going to be trying to cover that in about 45 minutes. The key that you need to know is that just pick 1 or 2 of these to remember and to be able to explain that there is intense complexity in design and these processes and these things. Now each person has 2 copies of DNA. One they got from their dad and one they got from their mom. And this goes for animals as well. Now each parent gave half of their DNA to the child. And then those chromosomes, those sections of DNA that they gave are then shuffled together, kind of like a deck of cards, so that their chromosome 1 and chromosome 2 don't match mom's chromosome 1 or chromosome 2 or dad's chromosome 1 the same way. They have all the same genes, but they're shuffled around. And then there's dominant and recessive genes. You can see in the top right corner the genes that basically control skin coloration. There's 3 main genes that do this. There is a dominant gene, a capital A, capital B, or capital C, and a recessive gene, A, B, C, lowercase. And depending on what section of these you have, if you have a capital A, lowercase b, lowercase c, you're going to have a bit more of a 3 kind of skin tone there, a bit middle ground. Now, if mom gives you a capital A, lowercase b, lowercase c, the left-hand column, and dad gives you the top row, lowercase a, capital B, capital C, that means that you basically could have as much as 6 as far as dark coloration, or as little as 0 as far as coloration. And this has actually happened. There was a group of parents, or a pair of parents, that had both skin colors about 3. And they had a pair of twins, 2 girls. One which was extremely dark in color, and one that was extremely light in color and ginger. Both came from the same parents in the same birthing process. This just shows how wonderfully intricate our design is, our DNA is the fact that we can get all of this diversity in a single generation. And that is probably how Adam and Eve was producing children. Because Adam and Eve would have been right in the middle as far as their heterozygosity, having half of each, having one high, one low, one dominant, one recessive, and giving to their kids the dominants or the recessives accordingly. And the same is for blood as well. Now, blood and skin both work a little differently. Blood, the dominant, is full on board, full control, while skin is more of a spectrum. So blood, if you have an A gene and an O gene, you're going to be type A blood. But if you have two O's, then you're going to be type O blood. So genes even then work a little differently. Everything works a little differently as far as our genes go, and it's incredibly complex, and the more we learn, the more just crazy that it is. But the dominant and recessive genes do hide the recessive genes a lot of the time, meaning that natural selection cannot act on them because they are not being exposed. The main process, the main designer from the evolutionary point of view, cannot act on them because it cannot see them. Does that make sense? All right. Next, we're going to be covering chirality. We mentioned this previously, that there is a right hand and a left hand orientation for all these things. And in a lab, when you produce amino acids or nucleic acids, then you get a left hand and a right hand version, 50-50. Yet, in life, we need the right-handed one. And there are 3 locations on every single DNA nucleic acid that it can be either left or right-handed, yet only one type works. So 1 out of 8 works. And then when you can try to connect them together, there are 4 different ways of bonding them together, yet only 1 works. So 1 out of 24 possible orientations work. And that's just 2 nucleic acids. The smallest and simplest life form is over 500,000 nucleotide base pairs. That means that it is 500,000 times 24 possible orientations, yet only 1 that works. This is an incredibly detailed coding system. And it's made even more incredible because we get 1 million DNA lesions a day. This we get from the water we drink, the sun that shines upon us, even the air that we breathe is damaging our DNA. Yet thankfully, we very rarely have any problems with this. Our body is remarkably well at repairing these. And occasionally it won't, and that's where cancer comes from. But without these repair mechanisms, we would die day 1 from all these millions of DNA lesions. And these systems are not only good, they are all redundant, meaning we have multiple systems that do the same thing, multiple different enzymes in those systems, and it's even systems that are special for each kind of damage. So this has amazing design characteristics in it. And it leads us to our first chicken or the egg situation. Because DNA repair is essential for life. If you do not have that repair, life will not survive. Yet the mechanisms for this repair are within the DNA itself, meaning you can't have one without the other. This is called Egan's paradox. We'll go over a number of other chicken and the egg scenarios. But let's get a little bit above the DNA itself, and this is something called epigenetics. This is a relatively new field of science, just a few decades old. Now, this regulates your DNA. So when the cell divides, you can see there the chromosome in the top left corner of that picture. Everything is tightly packed. It is a spiral within a spiral within a spiral, basically. It's incredibly compact DNA. And then below that section of organization, you have the histones. And this is DNA wrapped around these proteins. And they function kind of like those shelves that you may find in certain universities or libraries that you have to crank to open up. So there's only one hallway, but you're moving the shelves from left to right to get to different shelves. That's how these function. So you can see a pink gene there in the bottom center of that picture that you can't access because it's wrapped around that histone. So the histones have to unravel to access that. And it does that by certain things being bound on to that tail of it. And there's 15 to 20 different chemical modifications that can be done. And this shows which genes are regulated and which ones aren't. And to get this, you have to have the proteins, you have to have the RNA present to get these alterations. Yet, to get the RNA and protein, you have to have the genes expressed. So again, another chicken and the egg scenario. And these can help to hide a number of our genes and also last multiple generations. They have done different genetic studies where each year, each generation, different genes were expressed. And we see that by the phenotype, by what they're showing. And so you'll have a mother that smokes, and for the next 2 generations, their children's DNA expression will be different. So if your grandparents smoked, your gene expression, how you look, how you act, how you interact with things is different. How your mom ate when you were pregnant or she was pregnant with you changes how your genes are expressed. And this is just the tip of the iceberg. There are so many incredible things that affect these to affect our genetic expression, changing how we react to the environment. And it's just incredibly more complex when you actually look at how these genes are organized. Because when they're all wrapped together, all the genes that affect certain things are right there together when they're all rolled up. So if you wanted to access a certain area of metabolism because you ate a certain thing that's toxic, like chocolate, if we didn't have certain genes, that would be toxic to us the same way it is for dogs. Yet we can remove it from our system. And those genes are like in the same area, so that if the body wanted to activate it, the cell wanted to activate it, it would just send some proteins, some RNA to one area of the DNA to activate all the things that need to be activated. And it's extremely more complex than that, as we'll get into. So now let's look as far as what the genes do. So originally it was thought that one protein, one gene. So every gene was basically just a blueprint for a protein. That protein actually went and did the work. Yet it is wrong on both parts. We have genes that don't form proteins that do a number of other functions. And that was originally why we thought there was a lot of junk DNA, because it wasn't forming proteins. But we see that, at least biochemically, at least 80% of our DNA is functional, and it's likely that the rest 20% is as well. We just haven't been able to do those tests yet because of ethical reasons. But as far as the proteins go, not only does one gene make one gene can make a dozen proteins potentially. We have 23,000 protein coding genes, yet we have 300,000 proteins made from those. And that's basically by using exons and introns. You can see there in the bottom left. They take one section of DNA and they cut it up and splice it together in a different way to get a different protein. And by using certain exons and introns, you can get different proteins from the same section of DNA. You can see there in the bottom left, the bottom right, how one section can produce 3 completely different proteins. And that depends on what cell it's in. Sometimes it's the brain cell, sometimes it's the liver cell. Do completely different things. And this is made even more complex by the fact that that gene, the other side of the DNA, has to be read in reverse and still has a function. That is a reverse anagram, almost like a palindrome. And to be able to have billions of DNA letters do that, both forward and backwards, is just incredible things of design. That is coding that we cannot do. But now let's look at actually the proteins themselves. Proteins are essential to make proteins. So you get RNA coming out from the DNA. The RNA is then using RNA and protein to make proteins. Those are ribosomes, those are tRNA, and basically you're making a protein one amino acid, one block at a time. Yet all that requires proteins and RNA. So to get RNA and proteins, you have to have RNA and proteins. To get protein, you have to have RNA and proteins. So to get what you're trying to make, you have to have what you already made. Another chicken and the egg situation. And then proteins like to fold a certain way, and sometimes it's good, sometimes it's bad. Now we have these chaperone proteins that basically hold a protein in a certain formation until it gets to the protein that's going to fold the protein to be what it does to be a functional protein. Have I said protein enough? So it's just incredible, all the intricate things of design here to get a single protein. You have to have all this machinery already in place. And there's just no stepwise way of producing this. And it doesn't stop there. We're going to talk about protein just a little bit more. So to get a protein to where it needs to go, where it needs to function, you have to have delivery systems. And we have these little bitty legs here holding basically a sack of proteins. Now that is something called a phospholipid bilayer, and basically fat that circles in the protein and allows it not to just float off everywhere. And these tiny little legs, one step at a time, walk down this track. And thankfully they do it remarkably well, because these kind of things carry the proteins for our nerve to function. So if we want to feel something in our toes, we have to have these proteins there to react with the receptors. And then it starts a cascade that travels up the nerves very, very quickly. But we still have to have those proteins. And the only way to get it there, because the bodies of those nerves are in our back, or in our spinal cord. And those axons, these tendrils, go all the way down to our feet. So if you want to feel that foot massage, be thankful that these tiny little feet are walking, literally feet worth of your body down there. And these are microscopic little machines doing that. Thankfully they go very, very fast and do their job very well. But it's just incredible, the design in here and the complexity and the sheer scope of it. But enough about proteins, or we'll talk about it a little bit more. But now we're going to focus on something called a phospholipid bilayer. And you can see there in the top right, you have the heads, you have the tails, then you have the reverse tails and the reverse head. And so This is the skin of the cell. This is the outside border. This keeps certain things out and keeps certain things in. And these can form spontaneously in nature, but they're not the same as what we find in our cells. The ones we find in our cells are like that bottom right picture. It looks like all those balloons in the ball pit kind of picture. You have some on the top, some on the bottom, some that go all the way through. Those are all proteins doing a number of different functions. And they are actually so tight as to keep protons out. Those are helium atoms with just a positive charge. These can keep the smallest atom outside the cell. And it maintains this electrical voltage, which means the outside the cell is positive or inside the cell is negative or vice versa, depending on what's functioning there. And it allows certain things in and certain things out. This is incredibly designed, incredibly complex, and a sophisticated system here. And then this, of course, requires the chaperones that we mentioned before, and no cell can generate its own membrane, not from scratch. It can produce some, it can add some to it, but it has to have the membrane there that it gets from the previous cell when it divided. And so it's another chicken and the egg situation. You have to have the membrane there protecting everything, but you have to have the cell there functioning first. So which came first kind of thing? Is it the membrane, is it the cell? And so it's just another kind of chicken and the egg situation. And now we're looking at ATP. You don't even know what that stands for, just ATP is simple enough. There's a picture of it in the top right. This basically is the battery of the cell. And so when there's two little P's on there, that's phosphate on that little tail area there. That's an uncharged battery. And then the cell adds on a third phosphate there, making it into ATP from ADP. And now it's a charged battery. And it uses this for almost every cellular function. And we get this energy from food, from sunlight, etc. And phosphate is a very active molecule, meaning it's going to do a ton of things as far as interacting with nature. It's basically, as soon as it touches a metal ion, it's unusable for life. And certain things are unusable for life. That is why, for example, dog food, it's chemically tested. Well, in the 80s, it showed positive for magnesium in this case, I think. And dogs were eating it and they were getting sick because they couldn't access this magnesium. It was not bioavailable. The body could not use it. And so even though it chemically tested for there, just like a lot of phosphate will be chemically tested for, life cannot use it. Yet life needs this. Every life uses ATP. This is a very unnatural process, meaning it's not found in nature. And this little but very complex set of proteins there in the top and the bottom right is ATP synthase motor. This is an incredibly complex water wheel, basically. So you remember that gradient of the protons that we were talking about earlier in the cell membrane? It is using that gradient to charge these batteries. So the proton comes from the outside, goes in, and it moves the gears in this protein, which then allows the phosphate to be bound on to ADP, forming ATP. This is an incredibly advanced piece of machinery, and everything in life needs this. All life needs this. Yet, if you didn't have ATP, there would be no need for this molecule. But if this molecule wasn't there, if this set of proteins weren't there, then you wouldn't have ATP. Another chicken or the egg situation. Both are required to be present to be functional. And these are all examples of what can be called irreducible complexity. And that is the idea that when you take it down to the basic components where you cannot remove a single other component without the thing being not able to function, it ceases to function. The common example is that mousetrap there. You can't take a part away from that and still have it function as a mousetrap. Now, evolutionarily, first life could not be simple. I showed you a picture of the eukaryotic cell. The prokaryotic cell is a little bit less compartmentalized, but still extremely complex and has to do a number of functions all at once. It has to have been able to reproduce, meaning to split apart and divide. But it has to divide in just the right way. It has to have information on both sides. At the same time, it has to have the structure on both sides. If you have one without the other, it's dead. You also have to have enough energy on both sides. Again, without that, the cell is dead. It has to be able to eat. Yet what could it have eaten? There's no other organisms for it to eat. Photosynthesis is an extremely complex system, which we'll get a little bit into later. So it couldn't have been there present, just made things worse. but it has to have gained some sort of nutrients, some sort of energy somehow. And then it has to get rid of its wastes. Now, every biological system produces some waste byproducts, and the cell has to get rid of it just like we have to get rid of our waste byproducts. But evolutionarily, the first cell couldn't have been as refined as our current cells are, meaning it has to have been producing more byproduct with the same amount of intake, meaning that its excretory systems must have been even better than today's to maintain that. And that is incredibly complex again. So there's no real way that we can know of how they could have done this. These are all three big steps as far as trying to get evolution's first life that they have not overcome yet. These are big problems from that theory. Some other problems. We already went through how bacteria reproduce. They basically reproduce asexually, where you have one dividing into two, and 50-50 mix for the most part, and then those produce more and then divide again. And this is supposed to be how first life was reproducing. But this is a huge problem when we go from asexual reproduction to sexual reproduction, like we see with a lot of animals. Even when plants do this. So to have one, you have to have the other. Otherwise, it's no good. So if you have the male delivery system developed first, it's useless because there's no female integration system. Meaning you have a system here that is just a drain on resources. And natural selection should select for that out because this is a poorly developed system. It's not doing anything. It's just a drain on resources. Same way for the reverse. If you have an integration system, it should be being able to take things in and integrate it and be able to produce new offspring. Yet if there is no male delivery system, then that again is just a drain on resources. Now they try to say that it's a boon to animals, and it is. It does a number of things. It does promote genetic health, getting rid of mutations and maintaining good genes. But the problem is that just saying that it's good does not explain how it came about. That's not an explanation of where it came from, and this is a major problem for the evolutionary worldview. Now, photosynthesis. We're only going to touch on this a little bit because it would take a few chapters at least of a textbook in biochemistry. But it's basically the design complexity of a helicopter or a space shuttle. There is one part of it called photosystem 1. Now, this is in something called cyanobacteria, so it's not a plant, this is a bacteria. A prokaryotic cell. Extremely simple compared to a lot of other forms of life. Now, this takes 12 protein parts with a number of other cofactors. And these are just types of proteins and cofactors. There are a total of 417 pieces positioned in just so a way to actually perform its function. And these come from about 3 dozen genes. A relatively small number, but still a lot of information there. All this is within 22 billionths of a meter, so extremely small. Yet it has an antenna system, electron transfer chain, just incredible things of design that I'm not sure if we can even say that we've created a similar comparison on the macroscopic scale. So this is just one part of the photosynthetic system, extremely complex. And again, no stepwise way of producing this. If you remove one part, it does not function. Now let's look at some animals. So woodpeckers, I think Steve talked about this a little bit ago. They have to close their eyes when they peck, otherwise their eyes are going to be shot out. They also have to have glue on their tongue, so they stick their tongue in the little holes, and basically it has to attach to the insects. They then have to bring it in their mouth, and then they have to dissolve the glue. And they have the thickest skull bone mass of any living animal because they are pounding their head against hard wood. Yet if you have one of these, you have to have the other. If you have the eyes closing when they peck something to keep their eyes in, but that's not good to close your eyes when something's coming at you. It's not good to have that because then when a predator attacks, you can't get away because you're just closing your eyes. You can't see. Yet if they're pecking and they don't close their eyes, then their eyes are going to be out. The glue, you have to have the dissolvent of the glue, otherwise the bug is going to be stuck on their tongue. So they have to dissolve it and create the glue, but if you just have the glue without the dissolving, then it's just going to be a problem. If you have the dissolving without the glue, it has no use and should be selected for against from natural selection. Same for the bone mass. You shouldn't have all that thick bone on the skull because that's not good for flying. Thick bones are not good for flying. And yet, if you didn't have that, you'd be breaking your head every time you try to get food. Next are bears. Now, bears have their cubs just prior to hibernation, and they sleep for 5 months. And ask any mother, this is a bad idea to have a child and try to nap. It just doesn't work. Yet they make it work. And this is all while not eating, not drinking, and they only lose about 20% of their muscle strength. That's incredible. They get about half a gram of water from each gram of fat. They also recycle their urine so that they don't need to pee, and they retain all their water. So their urine is incredibly concentrated, but they still have all that water to use. And this is all while nursing these cubs. And if a human tried to do this, not only would they die from the lack of intake, but they would lose 90% of their muscle mass. And that's because the bears have muscle spasms to maintain the muscle mass. And all these are required to form hibernation. Yet, without hibernation, they don't really serve any purpose. Now, that being said, it would be wonderful just to go to sleep as a newborn, or with a newborn baby, wake up, and they're able to walk around, you don't have to worry about them, and everything's just taken care of 5 months later. That'd be nice. But to sum all this up, Job 12, 7 through 10 does a wonderful job. But ask the beasts, and they will teach you, and the birds of the air, and they will tell you. Or speak to the earth, and it will teach you. And the fish of the sea will explain to you, who among all of these does not know that the hand of the Lord has done this, whose hand is the life of every living thing, and the breath of all mankind. It's just incredibly complex, these design systems. You cannot produce them in a stepwise function. Maybe one or two you can try to get something, but the vast majority you cannot. And I don't think any evolutionist has tried, because they see that it is a hopeless task, yet they still try to find more information to prove their side. And now we're going to talk a little bit about natural selection. I'm sure some of you may remember these moths. You got the dark colored ones, you got the light colored ones. Back in England, supposedly, when the Industrial Revolution was occurring, you had all this soot covering the trees, making them dark. So instead of being majority white moths, the darker moths were then taken over because they could hide from the predators better, but the light colored moths were being eaten away. Now, they weren't driven into extinction. That gene is still in the population. But because when they landed on the trees, they were being selected for. And so the predators would reduce their numbers, so the black-colored moths were the more numerous. And some quotes from secular studies as far as natural selection. The most famous and influential example is Darwin's invention of the term natural selection, which he wrote in The Origin of the Species. Is daily and hourly scrutinizing throughout the world every variation, even the slightest rejecting that which is bad, preserving and adding up all that is good. Or, in short, evolutionary biology needs a designer substitute. Before Darwin, the ability to choose was largely confined to designing intelligence. That is, to a conscious agent that could reflect deliberately on the possible consequences of their choices. Or, the trick is, how do you talk about natural selection without implying the rigidity of law? We use it as almost an active participant, almost like a god. In fact, you could substitute the word God for natural selection in a lot of evolutionary writings, and you'd think you were listening to a theologian. It's a routine we know doesn't exist, but we teach it anyway. Genetic mutation and some active force choose the most favorable one. Yet, despite all this, they can't agree on a definition of natural selection. A quite general issue has still received no canonical treatment. What kind of thing is natural selection anyway? A law, a principle, a force, a cause, an agent, or all or some of these things? The present worry is that the explication of natural selection by appeal to selective breeding is seriously misleading, and that it thoroughly misled Darwin. What then is the intended interpretation when one speaks of natural selection? The question is wide open as of this writing. So, basically, it is being used as a all-powerful design selection process. The same way that humans were able to breed and produce certain breeds of livestock, dogs, cats, etc., natural selection is then using that same selective forces, same choosing forces on our environment, or the environment on living organisms, including us. So Darwin was the one that coined this term, and it was latched onto by the secular community very quickly, very strongly. But in short, genetic fitness tends to dominate which individuals pass their genes to the next generation. This is still a problem, though, because the likelihood of a newly generated beneficial mutation becoming established in an ideal population is twice the fractional fitness advantage. Since these advantages tend to be very slight, and since real populations are far from ideal, likelihoods for real populations turn out to be surprisingly small. Calculated as twice the fractional fitness advantage multiplied by the ratio of the effective population size to the actual population size, meaning that little formula there at the bottom, it has to be a very beneficial mutation to go from a single organism to the entire population. Or, in short, about 1 in a million. That's just for a single mutation, 1 in a million chance. And that's a beneficial mutation. But a lot of these mutations are not beneficial. In fact, the vast majority are harmful. Second, they're neutral. Very, very few times are these actually beneficial. And when they are beneficial, they're not really strongly beneficial. They're just mildly beneficial. So in short, natural selection is not strong enough to select for these things. And that's not even including all the recessive genes that we're talking about, all the epigenetics hiding things. So if creatures and humans' design share corresponding engineered features, how can we believe that adaptation mainly happens by random mutations and natural selection? That is anti-theistic because it's anti-design. This mutation selection explanation is harmful for science and theology because it misses all these things. If we try to address these biological problems from an evolutionary point of view, from an anti-design point of view, from an anti-theistic point of view, we're going to be missing a lot of things because we're not looking for the non-random. We're not looking for the non-broken. No rational engineer would use these processes because they are random. They don't work well. They're poorly at, they don't select things very well at all. They're not strong. They're not great at what they're supposed to do. And basically, it acts as a eugenics mechanism. If you have one gene that is slightly more advantageous here, like the coloration of the moths, then basically God is allowing eugenics to select out the ones that are not beneficial, the light-colored moths. And this changes how we can view God. As Gould said, it's perverse indeed. It mocks the goodness of God. So there is a new field of study called continuous environmental tracking. And this goes along with the epigenetics that we were talking about. But the fact that the biological systems are able to sense changes in the environment and change genetic expression, biological mechanisms. So our bodies, living organisms, are able to react to changes in the environment, changes in food, changes in weather, changes in a number of things, and then allow genetic expression to alter accordingly. And one such example is this fish. So many of you have probably seen these blind, no pigment cave fish. The evolutionary theory is that they got into a cave, and basically because skin pigment is an unnecessary thing, it was selected against. So all the genes, all the fish that had those genes died out because they were wasting energy. And all the fish that had this mutation that negated the pigment were able to produce. The same for the eye. The fish that had the eyes, it wasn't doing any good because it was completely dark. So they were selected for against because they were wasting energy. But the fish that got a mutation that prevented the eye from growing were selected for. Yet if you take this fish and put it in the sunlight for 32 days, the pigment returns. Those genes are still in there. Those genes just were shut off. And I wish they would have continued this experiment because I'm wondering if that eye would have come back in a generation or two. Because if these fish get out of that cave somehow, flood or something washes them out, they can go right back to functioning in normal daylight lit areas. For more information, you can look at icr.org/CET. They are the ones really pushing this. They're the ones getting into this. But it changes a number of things because the explanation of natural selection doesn't fit. Go back to those moths. Remember how they're supposed to be landing on the trees? They didn't. Those pictures, they had to glue the moths on. It doesn't fit. Their explanation is completely made up. They actually went back and tried to reproduce it. Couldn't do it a second time. They even had sections of England that stopped producing a lot of this soot. They went green and got rid of that. And yeah, the white moths came back in number. But the trees were still dark. Their explanation doesn't make sense as far as how the environment was supposed to be selecting against them. Yet this would. Maybe it's something in the pollution, maybe something in that we were putting into the air, causing a genetic change, causing a change in coloration. Not the genes themselves being selected against, but being turned off or turned on accordingly. This is a new and upcoming field of study. So if you are looking into going into anything biological, this may be something for you to further your career and also show the glory of God in His creation. So summary from this lecture. The fifth and sixth day is the creation of life. The creation of these cells, these multicellular organisms, these living creatures, incredibly designed. To get these from nothing does not make sense. There is no evidence for it. Abiogenesis is no different than spontaneous generation, things coming from nothing. The Miller-Urey experiment has been disproven in multiple different ways. Information science, the fact that we have all these coding systems in our DNA, and it is more complex than any computer coding. I wish I could go into detail, but honestly, I don't even understand it enough. But there are geneticists out there that can do a wonderful job, at least trying to explain it for me to understand, and you as well. There's just incredible complexity in this. The design is so apparent that they have to deny the design. They say it's apparent design, not really design. Don't trust your eyes. Natural selection is no replacement for God as far as His control over life. He has made us so well designed that things are able to turn on and turn off depending on the organism's needs. Continuous environmental tracking is a much better way of looking at things biologically. It is not anti-design, it's not anti-theistic, but purely theistic. It is showing design in everything. All living things have this. And it's just incredible what we're learning the more we learn about life. For some resources, there's not as much because a lot of this is in biological textbooks. You'll just have to read those if you want more as far as the complexity. It's there. I honestly don't know how a person can go through biological classes at college and not see this apparent design. It just is right there in front of your face. But for some more as far as walking through some of these arguments, here's some information. Thank you.